Good evening, and welcome to the Professor Andrej Župančič Memorial Lecture at the Synapse Neuroscience Conference 2021. At every biennial SNC, there are fewer and fewer of us who have had the opportunity to meet Professor Župančič in person, or even had the honor to be in his circle of friends. So it kind of falls onto us in the old guard, if I may use that term, to let the others, younger ones, know just how exceptional he was. I will just use a minute to present him in a few succinct words that don't do him justice. But I would say he was a physician, a pathophysiologist, a neuroscientist, a teacher, an ecologist way before it was fashionable to be one. He was a video documentarian. He was an avid mountain climber. He was an explorer by heart and a profoundly curious innovator. It might surprise even some of the people who knew him that he actually authored a Nature paper in 1965 that was entitled Creation Rate of Matter and the Heisenberg Uncertainty Principle. That is a subject quite remote from a typical topic of a neuroscientist. So his friends and students are devoting a special lecture to his memory at every Synapse Neuroscience conference. And this year, we have a very special speaker and topic. Dr. Sonia M Vallabh holds a BA from Swarthmore College, a JD from Harvard Law School, and a PhD in biological and biomedical sciences from Harvard. As you are about to hear, her work is a personal mission. Dr. Vallabh works at the Broad Institute in Cambridge, Massachusetts, the United States, where she co-leads the initiative to develop treatment for prion diseases. And today, she will share with us her story entitled Genetic Prion Disorders and Patient Scientists' Mandate. But before we shine spotlights on Dr. Vallabh, I would just like to add two more things. I would like to thank our sponsors, IBRO Pan-European Regional Committee and the Slovenian Research Agency who supported this event. And I would especially like to thank Radio Slovenia, third channel, Ars, for joining in and streaming the talk and the evening conversation with Dr. Vallabh. In regards to the conversation, it is going to follow the presentation by Dr. Vallabh. It is entitled Neuroscience and Society Dialogue Event, and society can and is most welcome to participate by posing questions for Dr. Vallabh to answer. To do so, you can go to our webpage and pose the questions using the Slido application. This is all from me for now. Dr. Vallabh, the stage is yours.
Thank you so much. I will quickly bring up my slides. And I'd just like to say, in the meantime, that it's an honor to be here. Thank you so much for the invitation and to be presenting this memorial lecture. As you've heard, I will be talking about my scientific journey, but it also comes from personal experience and a personal motivation. So I'll start my presentation there. This is a photo of my husband, Eric Minikel, and I getting married in 2009. And it was a beautiful wedding. It was actually in my parents' backyard in Pennsylvania. And this is a photo of my mom helping me get ready that day. So she was the person who organized the whole event. She was 51 years old, and at this time, healthy, and on that day, very happy. And we couldn't have known that this was actually the last major family event for which she would be with us. So this was August of 2009. And it was only about 6 months later that this person who had, single-handedly orchestrated a wedding began to mysteriously and before our eyes lose her abilities. It was very hard to explain what was sort of unfolding before our eyes, but from day to day and from week to week, she went from being able to, communicate and clearly with us and live independently to being confused, being unable to finish a sentence, being unable to get around the house independently, becoming so weak that she couldn't stand, becoming unable to feed herself, unable to sort of recognize us. All of this happened over the course of 4 months. And we rushed her around to hospitals in a panic, but despite having blood drawn, I want to say hundreds of times, despite having her spinal fluid tested over and over, despite having her brain imaged so many times, nobody could tell us what was happening. She went onto life support that summer of 2010, but we still had no diagnosis. And so it was very hard to reach a family consensus about what to do. And it was only at the end of that terrible year that we got a preliminary diagnosis that maybe this could be prion disease. And based on that information, my family was able to reach a consensus to take her off of life support, and she passed away 2 days before Christmas. So that was a totally unanticipated year and a brutal year for my family. But what came next was even more unexpected. We received her autopsy results and learned that this had not just been prion disease. She had actually died of genetic prion disease. And this reopened the whole terrible chapter that we thought we had just closed. To us, this meant that the story wasn't over. And actually, I was at 50-50 risk of having inherited the mutation that had caused my mom's disease. So the question was posed to Eric and I. We're now in late 2011. You're at 50-50 risk. How would you like to proceed? And we were told by many people, you should consider walking away from this and foregoing predictive genetic testing, because this is a fatal and untreatable neurodegenerative disease, and there is simply nothing you can do with an unlucky result. We talked to physicians, we talked to genetic counselors, we talked to friends, we talked to family, and I would say this was really universally the counsel that we received. Nevertheless, we sort of knew from those very first really gut-wrenching moments that even though this was a terrible situation to be in, there was no way we could sort of get out of it by ignoring it. And for us personally, it felt that now that we knew about my risk and that was going to be a part of our life, we at least need to know more specifically what we were up against. We needed to get tested. And so we set about the process, and it was December of 2011 that we went to Brigham and Women's Hospital together to receive my test result. And this is the piece of paper that we were handed that day. So it was not the result that we hoped for. I did inherit the disease-causing mutation from my mom. So what did this mean to us at the time? We knew that this mutation causes a fatal, untreatable neurodegenerative disease. In fact, we had just seen it happen. And this mutation and a handful of others in this gene do have a very high likelihood of causing disease. That was known at the time, and we've done some more work in that area since then, kind of confirming that these are very high penetrance mutations. In the meantime, the tempo of disease is unusual in the sense that carriers remain completely healthy for a variable number of years, but once symptoms begin, the disease is incredibly rapid. What we saw with my mom is actually quite typical. From first symptoms to death is usually under a year. So these are the cards we were dealt. And at the time, we were not scientists. We were not particularly well equipped to receive this information. So I had trained as a lawyer. Eric trained as a transportation engineer. And we sort of went home with this test report and realized that this sort of new domain was opening up in front of us, and we needed to be able to understand it. So this started as sort of a very humble self-education quest. We went online, we literally Googled the word prion. We started trying to understand what is known about these diseases and who's working on them. And, part of our motivation in those early days was just the diagnostic odyssey that we had been on with my mom that was never successful in her lifetime. I think that really conditioned us to believe that prion disease is sufficiently rare that doctors won't recognize it, and you can't count on them to know what if there's progress in the next several years? What if there's something that actually I could be doing, or some medication that I could take at some point in the future? Well, we just need to be able to identify that ourselves. We need to be savvy consumers of scientific information. So that is where we began. And from that starting point, a major life change was sort of catalyzed. We actually ended up leaving our jobs as the process of re-educating ourselves drew us in. We took night classes around Boston. We took new jobs, sort of entry level in science at MGH. And we started to try to get a handle on what does it mean to do research every day? And is there some way we could meaningfully contribute to, in particular, the development of a therapeutic for prion disease in our lifetimes? That question led us to a PhD program, it led us to Harvard Medical School, and it led us to the Broad Institute. This is where we did our PhDs, and it is also where we have based our independent lab since defending our PhDs. And we've just been beyond fortunate to land here, because I think our particular problem has found a lot of uptake here. We found mentors who were willing to bet on us, even when we were very new to this, and who were willing to advise us on our quest, even as it started to expand. And we realized that the number of problems that we have to solve is more than just what is the molecule going to look like, what is the drug going to be. So we're very fortunate to be here. And this is a little bit of a snapshot of what our life looks like today. So our lab is based at the Broad. We're also instructors at Mass General Hospital. And we are doing everything we can to sort of get prion disease on the radar so that affected patients and families can find us, and so that we can rally a team around this rare disease and really try to do something about it. So what I will try to share today are some of the puzzle pieces that we have realized have to come together if we're going to make progress on prion disease. And as I said, it's more than just finding the drug, although that itself is a big challenge. But I think in a rare disease, we also have other challenges. So we have to really pin down who's our patient population. We need natural history on what the disease looks like. We need molecular tools that will help us understand if the drug is working. And we need a plan for what to do in the clinic, how to meaningfully test a drug that we might find or help to identify. And this last piece, sort of what the clinical plan looks like, I think for any rare disease, it's probably going to be a little bit tricky to get that part right. So for us, figuring out that plan has meant talking to the Food and Drug Administration early and often, and trying to carve out a path that will satisfy the level of evidence that we all need, while also honoring some of the specifics of this really difficult disease. So as I talk through some of those aspects of our research program, I also hope that I can sort of work in this particular theme that has made our lab and our work unusual from the very beginning, which is we're patient scientists, and we simply look at the disease differently, I think, than people who haven't watched it unfold in their own family and who aren't personally at risk. One big thing that we have sort of like elevated to the center of our mission is prevention. We've realized that we don't just want to treat people once they're sick. I think in some cases we won't have a choice because we won't have the foreknowledge that someone is about to develop prion disease, but in some cases we do. So how can we leverage that predictive genetic testing information towards trying to keep people healthy longer? In the meantime, there are so many amazing biotechnologies sort of coming up today, many of which I hope will have a big impact on human health as the decades unfold. For us, we've got this very particular lens on the nearest term practical solution. We need something that we can not just sort of devise in the lab, but actually apply in the clinic within, ideally, the next several years. And that really pushes us in a different direction than many people in the academic sphere. Finally, we can never forget that our most important constituents are prion disease patients and prion disease families. They are sort of supporting us every step of the way in various ways that I'll talk about. They participate in our studies, they donate to our research, and we owe it to them to stay focused on our mission, but also to share information that bears on them, maybe earlier and more often than a typical lab would. So with that, I'll give a quick introduction to prion disease. You may be familiar with the term CJD. Prion disease in the clinic goes by a range of different names. And as of today, we really think of these, Eric and I, as sort of historical accidents. Prion disease can also come about sort of uniquely through 3 different etiologies. So about 15% of cases are genetic, like what we saw in my family. Most cases are sporadic, meaning no known genetic or environmental trigger. They just seem to happen. Rarely, but very famously, cases can be acquired. Luckily, that is vanishingly rare today. There's also a lot of diversity in what the early symptoms can look like. Basically, any domain of brain function can be the first affected, although you rapidly get sort of progression towards this rapid dementia. But despite all of this sort of alphabet soup and all of this diversity in how prion disease can come about or how it can present in the early days, I think we have really benefited from this blueprint that at its heart is very simple. So all prion disease really follows this one pathway. We have a single gene, the prion protein gene, that encodes a single RNA that encodes a single protein. And here in this sort of friendly blue cartoon, we're looking at the normal prion protein, which is not a problem. We all have the normal prion protein. We express it basically in all of our tissues. And in the course of a normal lifetime, it will never cause you a problem. However, it is capable of misfolding into this post-translational conformer, this red sort of misfolded cartoon that has the ability to grab other PRP molecules and misfold those as well. So in this way, misfolded prions can sort of trigger a domino effect across the brain. Normal PRP keeps getting converted and misfolded at this amplifying rate. And for reasons we don't totally understand, these prions kill neurons and lead to neurodegeneration and ultimately death. So this blueprint has really oriented our goal from the beginning. And prion protein sort of sits in the middle of this pathway as the pivotal molecule in the disease. The goal of our lab is to find safe and effective ways to lower the amount of PRP in the brain. And we are actually really lucky that we inherited a lot of data suggesting that this should be an effective strategy. So the way we model prion disease in mice is that we can actually inject regular wild-type mice in the brain with prions, and they will go on to develop full-blown fatal disease. That usually takes about 5 months. If that animal is, instead of a normal wild-type mouse, a transgenic mouse that expresses more prion protein than normal, that whole process will unfold faster than normal. If you delete one copy of the prion protein gene, so that animal now has 50% of the normal level of prion protein, it'll take twice as long to get sick. And if you delete both copies, so the animal has no prion protein in its own body when you inject it with prions, it will be completely protected from prion disease. So this suggests that without that fuel to the fire, without that normal PRP that can be converted, you really can't initiate prion disease no matter how hard you try. In the meantime, we have several different lines of evidence that lowering the amount of PRP in the brain should not in itself be harmful. So we have a variety of different animals that have been engineered to lack prion protein, and they've been characterized for years, and no severe health outcomes have been identified. These appear to be healthy animals. We led a study a couple years ago where we were able to identify for the first time people who are walking around who happen to have one inactivated copy of the prion protein gene. So we can think of these people as experiments of nature, and those people are healthy too. So I think this all suggests that we can dial down the dose of prion protein without causing other health problems. So that all brings us to the question of, if we want to lower prion protein in the brain, what is the near-term most practical way to do so? And around 2014, we first struck up a collaboration with Ionis Pharmaceuticals to look at antisense oligonucleotides, or ASOs, as a technology for lowering the amount of one disease-causing protein in the brain. So some of you may be familiar with ASOs. They've actually gotten quite famous since then. But these are short, chemically modified strings of DNA that sequence specifically bind a target RNA. So in our case, we'd be looking to design an ASO that can specifically bind the RNA for prion protein based on its sequence. And that binding event triggers both to be cleaved. So you're sort of cutting up that target RNA, reducing its levels, and reducing the amount of protein that's ultimately made. What was especially attractive to us about ASOs is that they were starting to accumulate data in non-human primates and in humans for ability to deliver, sort of distribute around the brain, and get around the brain following delivery into spinal fluid. So one of our greatest challenges, and this is true across neuroscience, is the difficulty of getting a drug to the brain. And in the case of prion disease, this is really a whole brain disease where we can't just inject focally into one particular region. We think we have to reach the whole brain. So the idea that ASOs could be delivered by lumbar puncture into spinal fluid and then reach sort of across the brain, not completely evenly, but still reach all the regions we care about, was very attractive. So we struck up this collaboration, and scientists at Ionis sent us sort of preclinical tool compounds. So what you're looking at is data from mouse ASOs that target the mouse prion protein RNA and reduce its levels in the brain by about 50%. And it was in our court to take these molecules and show, do they help in a prion disease animal model? So I described this model earlier. So we inoculate the mice with prions, and they sort of replicate prions and then develop really a spookily reminiscent disease. It really mimics the symptoms and pathology of human disease very closely. And here's the survival curve out of the first study that we ever did in our animals here at Broad. There's much more data that we've generated since then, but I think this first plot really shows that the active ASOs, 1 and 2 in blue and green, cause those mice to outlive their control counterparts by about 60 to 80%. And we validated this result with a second set of ASOs, slightly different sequences, slightly different chemistry, and again saw a comparable extension of survival. In the meantime, we did a validation experiment at a second site, our sister lab at NIH, and they also saw this fairly dramatic extension of survival. We wanted to ask, how much do you have to reduce PRP to see a survival benefit? And so we did a dose response experiment and found that even the lowest dose we delivered, which corresponds to a 21% reduction in prion protein RNA in the brain, increased survival measurably by 15%. So the more we lower, the more good we do. But this does look like a continuous dose response relationship. And with that early treatment, this was a dose dependence not just in survival, but in maintenance of body weight, in time to accumulate symptoms, and in maintenance of normal nest building activity. So here we started to see that early treatment was actually extending healthy life. With yet another set of collaborators, we did some experiments where we let the disease go a little longer. We waited until there were signs of pathology in the brain and then gave a single dose of ASO. and we're able to bring those signs of pathology back down. So here we started to see that even with sort of established molecular disease, an ASO could be helpful. We also tested across a range of different prion strains. So misfolded prions can come in different flavors, and saw that across strains, lowering this common substrate of prion protein was effective. So this gave us a lot of hope for this general strategy, because prion protein is essential to all of the different versions of prion disease. Maybe this strategy can be helpful for all of them. Finally, we wanted to really drill down on what is the impact of treating at different stages in the disease. And here what you're looking at is a chronic dosing experiment where we dosed the animals every 90 days as long as they survived. The long blue dashes, these animals started getting treatment early. And by early, I mean up to the point where neuropathology is detectable in the brain. If we dosed up to that point, we could actually triple the survival of these animals. And it was about as effective as deleting one copy of the prion protein gene. Now, in the small blue dots, these animals had advanced neuropathology and they were on the cusp of symptoms. And you can see that we still had an effect, but the effect was much less pronounced. So based on this, we pushed even deeper. We treated mice that were actively symptomatic. Every mouse had lost weight. You look in the cage and you can tell that they're sick. At that stage, we could prolong life for about a third of the animals, but not for all of them. And if we pushed even deeper to a point where some of the animals had already reached terminal endpoint and we treated the remainder, this was too little too late. So what have we learned so far? ASO treatment that lowers PRP appears to work. It's dose dependent, it appears to work across strains, and it appears to work across disease stages to a point. But there is a distinction between what we can do early and what we can do late. And that brings us to the idea of enabling prevention as we look towards the clinic. So we have two patient populations in prion disease, symptomatic and pre-symptomatic. And for symptomatic patients, trials have actually been done. They didn't work. These were two different small molecules that didn't have strong preclinical data behind them. But I think these trials have shown us that you can recruit for a randomized placebo-controlled double-blind trial in prion disease. We have certain assets. We have a very good diagnostic assay now that can almost definitively diagnose prion disease from a CSF sample. We have a well-established surveillance network. And the fact that the disease is so rapidly progressive actually means that you can use a survival endpoint. There are challenges here as well. By the time you recruit people to a symptomatic trial, they're in many cases severely debilitated. And in one of these trials, in the doxycycline trial, about half of the patients recruited were already on life support. So that is a major challenge for figuring out how much good we can do for people at that disease stage. But I think in terms of having a playbook for doing a symptomatic trial, the resources exist. The big challenge is just to push diagnosis earlier. For pre-symptomatic patients, the landscape is a little more complicated. This is not usually the population in which we do clinical trials. These are healthy people. We have done some work to identify the high-risk mutations. So we know who is at high risk of developing the disease at some point in their life. We've done some work to characterize age of onset. So we have a sense of the distribution for each mutation when people get sick. And we've created a registry to try to recruit people online so that we have a sense of where carriers are and who is interested in research and trials. But I think we need more. We actually need a totally different clinical plan for once we get carriers enrolled in a preventive trial, what will we measure? Because if they're already healthy, we can't just treat and see who feels better, who functions better. In the meantime, we actually can't randomize to drug or placebo and then follow all the way to onset. And part of the reason is that between the rarity of the disease and the variability in age of onset, we actually would not be able to power a trial of that design. I also think we would not be able, as a rare disease, to convince a pharmaceutical company to bet on such a long-term trial. So what we need instead is a biomarker. We need a plan that's built around a molecular measurement, something we can measure in healthy people that will tell us if a drug is working or if it's reasonably likely to benefit carriers. So with that in mind, we launched a biomarker study in 2017 at Mass General Hospital, and we've been bringing in genetic prion disease mutation carriers and controls. Some participants have now made up to about 5 annual visits, and we've been looking at biomarkers. And I'll share at a high level some of what we've learned so far, although the study is ongoing. We've looked at neuronal damage markers in these carriers because these neuronal damage markers, for example, neurofilament light, go up very dramatically in symptomatic disease. We also know that in slower dementias like Huntington's, neuronal damage markers are going up years or maybe even decades before symptoms. But in prion disease, when we look at our carriers and our controls, they actually look the same and they're all in the normal range. So this suggests that it's not the case that neuronal damage is accumulating gradually over decades in prion disease. We've also tested the RT-QuIC assay on CSF from these individuals. So RT-QuIC is the diagnostic I mentioned earlier that can detect prion seeding activity, even at very low levels. And you can see in red that for symptomatic CSF, it's a very robust assay. It gives you a very strong signal. But when we look across our carriers, for the most part, almost all of them are negative for RT-QuIC, suggesting that prions have not yet formed in these individuals, not at a level that is measurable in CSF. So what can we say so far? Now, the study is ongoing, but I think it's really powerful that so far our data suggests that most carriers are healthy for the vast majority of their lives. I think many academics would call this a negative result, and they would say, how disappointing, you invested all this effort and you didn't find anything. I actually think we found the best thing. We found that these people are healthy, meaning maybe we have a long opportunity to keep them healthy. And I think these are data that we owe the community. So, I think in a natural history study like this one, some labs might go decades without publishing anything. We published these results after just a couple years. And part of our motivation was to get the message out there. I think this is meaningful to carriers. It's meaningful to the participants who so generously gave their time, their body fluids, right, their effort and energy to our study. And it actually impacts how people see themselves. I think knowing that you don't have an incipient disease process in your body, that's powerful. Unfortunately, in prion disease, we encounter some medical discrimination as well, where people think that people with prion disease or at risk for prion disease are a biohazard. And sometimes people are denied necessary medical care. So I think from that perspective too, if we can show that people are healthy for decades, I think that's really important. But these data also have a direct impact on our clinical strategy, because we're probably not going to be able to do quote unquote secondary prevention, where we recruit people who already have some sort of subclinical signs of disease and then treat to see if we can reverse those subclinical signs of disease. If our carrier population is healthy, then we need to do something even bolder, which is say, these people actually seem completely healthy right now, but based on a genetic marker, we're going to go for primary prevention. We're going to treat them to try to keep them completely healthy. From that perspective, here's something else we've learned from our biomarker study. Prion protein levels in CSF are nice, steady, measurable. We can look at the same person 2 to 4 months apart, and their prion protein levels in CSF will be quite similar. And that's true whether you're a control or a carrier of one of these various mutations. Why does this matter? If our therapeutic goal is to reduce the amount of prion protein in the brain, maybe one way we can show that we've done that is by showing that we've lowered prion protein levels in CSF. So if we want to show we've lowered it, having a nice steady baseline is really important. And we've been able to follow people out more than a year now, and we've seen that that baseline is maintained within an individual. So conclusions so far, CSF PRP seems like it could be very informative for a trial. In the meantime, we're not probably going to use signs of prodromal disease to sort of design and orient our trial. We have an opportunity, and I would argue a mandate, to shoot for primary prevention. So what does that mean as we project forward? The clinical strategy for trials in carriers that we have proposed looks like this. We go back to our very simple blueprint of disease. We use a drug that will lower either PRP or one of its precursors. So in the case of ASOs, we lower the RNA in the brain, and then we measure prion protein levels in CSF. And could that measurement, it's really a drug activity biomarker, a pharmacodynamic biomarker, but could we elevate that to the primary endpoint in a trial in carriers? Could we elevate it to a basis for provisional approval of that drug? Because we are so confident, just based on everything that's known about the biology of the disease, that lowering PRP levels should predict clinical benefit in these individuals. So I can tell you this is not business as usual, but we took this plan to the US Food and Drug Administration in 2017, and we sat down in a room with 25 FDA scientists, and we laid out why we need prevention in this disease, what the disease looks like once it strikes, how little time you have. And, we have every reason to think as of right now that the function that people lose on that dramatic downhill slope is going to be much harder to rebuild than to preserve. So we made our case for prevention, and we also laid out the clinical path, the clinical plan I just showed you, proposing that we do have an informative biomarker that we could treat towards in a healthy population. And what we heard from FDA scientists that day was, this is ambitious, but this is also a really important problem. And it's going to take a lot of data, but we're willing to work with you to make sure that the right data are gathered, because we think that there should be a path forward here. So they were supportive, which was really, for us, amazing. And we have since had more detailed interactions in which they have indeed provided detailed guidance on all the data we need to gather. It is a lot more homework than if our sole clinical plan was to go into the clinic and treat symptomatic patients in a more standard clinical trial paradigm. But we have set about trying to really nail down the relationship between the drug and the target and the biomarker and the disease outcome across multiple different models, multiple disease stages. We are trying to pin down the biomarker as closely as we can. We created a new assay. We've measured it in multiple different ways. And we're continuing to push forward natural history to make sure that we've characterized our trial population as deeply as we can. So all of these are sort of ongoing fronts in our lab. And where does that sort of leave us for now? Ionis has said that they hope first-in-human clinical trials of PRP-lowering ASOs could launch in 2022. And they are aiming for both a symptomatic and a pre-symptomatic clinical path. So we're sort of hustling to make sure that the sort of ducks are all in a row for that pre-symptomatic path to be viable. Hopefully within the next year or two, that process is underway. Slightly larger picture, I think it would be interesting to see within, say, 5 years, if we can take everything we've built, the natural history and the biomarker, the clinical path and the regulatory engagement, the proof of concept, and can we leverage all of that towards somehow lowering prion protein even more? And will that be with a sort of new and improved next generation ASO technology? Will it be with a different technology? I think we have convinced ourselves that lowering PRP is effective, and the more we lower, the more good we can do. So if we can dip below that 50% that we've achieved in the preclinical studies I showed you, how much more good can we do? That is an open question. Finally, in the very big picture, we are a small disease. Prion disease affects 1 in 6,000 people roughly, and genetic prion disease is only 15% of cases, so yet a smaller number. But I hope that if we're able to drive forward a primary prevention paradigm, that could actually be meaningful beyond our one disease, because we're not the only disease that will be better treated by prevention than by waiting for things to go wrong. And I mean, especially in neuroscience and especially in neurodegeneration, we're talking about a tissue that we understand so little about still to this day and have very limited ability to rebuild. So I hope that we can provide one example of how might we go about trying to do rigorous and meaningful drug development to keep people healthy. Finally, we sort of walk this divide every day between trying to bridge what patients want out of the research ecosystem and what biomedical research rewards. And it's tricky. Sometimes it feels like we're kind of balancing different identities as we think about having to apply for grants and having to write papers and having to make sure that we can sustain our lab by traditional means in order to keep doing the work that we do, but always without losing sight of the fact that this is for a reason and we have to get to meaningful therapeutic as fast as we can. I hope that somehow we can help pull those two worlds a little closer together, but it's not easy. In the meantime, I will just end on a note of optimism. Early on, folks told us there's nothing you can do. And I don't think anybody meant that to be, discouraging. I think they were trying to protect us from getting our hopes up. We've really found since then that we're surprised by how much we've been able to push forward. And of course, there are no guarantees. But even if we hadn't pivoted to working in science and to running a prion research lab and to working actively in drug development ourselves, one other thing was available to us because we learned about MyRisk, and that was IVF with pre-implantation genetic diagnosis. So I think many of you will be familiar with IVF, the fertility treatment. It's possible now to create embryos and then do a tiny biopsy on them in vitro and identify whether they carry a disease-causing mutation like mine or not. We went through this process and it was many years of our life and it was very far from easy. But we had a miraculous outcome, which is we were able to identify healthy embryos, and those were transferred back into my body, and I was able to achieve 2 healthy pregnancies. One in, let me see, Daruka was born in 2017 and Kavari, our son, was born in 2020. And we have as much confidence as it's possible to have that they are mutation negative. So I think optimism comes in very different forms. And in some ways, this is the ultimate form of prevention. And we've already had the tremendous good fortune to do it. So with that, I'd just like to say thank you so much for having me and for your attention.
Dear Sonia, I just read a commentary. Somebody said, I wish I could clap. So basically that just sums it up for this moment. I would like to thank you from the bottom of my heart for the presentation. And now I would like to dive deep into whatever the stream of questions we've collected in the meantime.
Absolutely.
So if it's okay with you, we would have a short break and resume at 10 to the hour.
That sounds great.
Okay, perfect. I encourage everybody who is listening to us to maybe, if they haven't already done so, go to our webpage and use the Slido application to submit a question. See you back in less than 5 minutes. They don't go into too much detail, and the other ones deal with a broader picture. So where would you like to start?
Oh, I'm happy either way.
Either way. Maybe from the perspective of a rather unique perspective of someone who had actually at 27 years old had to decide about the profession. You've obviously chosen law initially because I suspect some affinity for the field. And then when you suddenly had to reframe the way you look at the world altogether and choose a totally different path, that must have required overcoming quite a number of obstacles, because apparently science wasn't your first choice before. What were the obstacles that you faced in the change of direction at the beginning?
Well, I mean, there were quite a few. I would say that I had never considered becoming a scientist. I had my father is a physician, so that is something that had always been on my radar, but research science had actually never been on my radar as a kid or growing up or going to school. And it was really just when this happened to us that this dichotomy became very clear. We had spent all this time talking to doctors during my mom's illness, and the doctors were saying, we have no idea, or like, now we have maybe some idea, but there's really nothing. And then a few days, it was maybe 2 weeks after we got my genetic test result, a friend came over who is he's a scientist, he's a chemist and materials scientist, and he came over with like a thumb drive, and he said, you should know that there are people working on this. And he had just gone on to PubMed and pulled a bunch of articles. And my initial reaction was, no, like, this is too rare. There's nobody working on this. But sure enough, he had found some articles, and that sort of pulled back the curtain for us on the fact that even rare diseases for which currently there's nothing in the clinic might have a research ecosystem behind them. And once we figured that out, that seemed like the place where we needed to be. But the process of getting there was, it was incredibly painful. I think in the beginning, we spent a lot of time just trying to ingest everything we could find, trying to read articles, but of course they're super jargony and it was super slow, and walking into classes, and we like cold called researchers, and we just tried to get people to talk to us. We were lucky to have scientist friends who came over to our house and sat down with us and talked us through things that we couldn't understand and helped us to start to sort of build a vocabulary. But I'd say the bigger challenge was not building the vocabulary to sort of ingest the research that was out there. It was building the sort of lens to figure out what was going to be useful to us and what wasn't. Because there are just thousands and thousands and thousands and thousands of papers on prion disease. And there are a good number of sort of therapeutically oriented studies that end with the words, this is very promising and deserves future study. And then you look at the date, and that was in 2002, and there hasn't been any future study. So I think we had to orient ourselves around the biases that lead to things like what's published and what do you say about it, and how do you frame your findings, and that might not always point you in the direction that things are actually going to move in the future. figuring out what was practical in terms of a therapeutic, what could move faster versus what was, maybe promising but very blue sky. That all took, I think, years. Just being able to sort of prune the tree of different possibilities. When we first came to Broad, we had a lot of very different ideas about targeting the prion disease process at different stages. And we were interested in, can we target misfolded prions? We were interested in, can we target neurotoxicity? But those are parts of the process that we just don't know that much about right now. And I think it took us a while to get our sea legs and understand, actually, we're probably 10 years or more, 10 or 20 short of the basic science that would enable those drug targets. And we've really got to focus on what's available to us. And that's kind of what brought us to focusing on prion protein itself. So there were definitely, I would say that was the big intellectual obstacle, was just developing the ability to sift through everything, to find the experiments that were worth us investing in and trying to carry forward. I think in terms of like logistics, we were very fortunate that we didn't encounter as many obstacles as we expected. And I think we're very lucky to live in Boston, where there's just so much research, and we were able to find these sort of entry-level jobs, and we were able to find people willing to take a risk on us. And, sort of all along the way, like when we applied to PhD programs, we were very upfront about why we were making a career change and what we hoped to do. And that didn't resonate with everybody, but we found advocates who thought, these people are really motivated, so probably they're gonna go out and work hard and try to make this happen, and who were willing to sort of bet on us. I think we had initially imagined that it simply might be, we might encounter roadblocks to making the career change that would just make it impossible. And instead, at each stage, we thought, well, this much was possible. What if we push a little further? What if we push a little further again? By putting ourselves out there, we realized that more was possible than we would have guessed.
Yes, I mean, I hear you. I understand the perspective. I still think it's obviously an amazing mixture of the specific characteristics that the two of you have that somehow landed on fertile ground of so many other circumstances being favorable enough to make the progress possible. But as a scientist myself, who has issues with the academic incentive structure, with issues that you have touched upon in your presentation, for example, reluctance to publish negative data, and how valuable it would be if we could somehow restructure that whole system by which science is currently used to running. Given the situation in which we find ourselves these days, globally speaking, about some sort of a crisis of science reputation, what would you as a scientist maybe suggest your fellow scientists to try to do in a systematic manner to improve our collective act to make larger strides in terms of progress in translation, but also doing better science day by day?
Yeah. Gosh, that's really tough. Yeah, I think the incentive structure is, I mean, it's the case in any profession, right? That you end up with incentive structures that don't perfectly align with the mission that brought you there. And I think when we came in, we had like kind of a fierceness that came from being new and being from the outside, where we thought, well, we'll just do things differently. And in some ways it's been possible. In some ways we've definitely had to compromise. I think Eric writes a blog, cureffi.org, that has been active since we got into science. And it was really sort of a forum for just speaking out loud to anyone who would listen and trying to share our ideas and get feedback and iterate quickly. And I think we had a vision in the beginning. What if everything's like that? What if all the data we generate is just open on the internet, like as soon as we produce it? Can't we sort of break out of the regular system because we're in a hurry? And the answer ends up being yes and no. Like, I think things have become more complicated as we have, and we're very fortunate that this has happened, as we sort of accumulated more collaborators who, have all sorts of needs in this environment professionally, and it is really important, for their career that they have this data not as a blog post, but as a first author paper. And it is really important to our pharma collaborators that they be able to, protect certain IP, because that is what will allow them to financially advance the program. And so, we have had to sort of like re-strategize around all of that. With that said, we do try to make as much data as we can public when we can. And with our ASO studies, like individual animal level data is actually available online in our GitHub repository. And I hope that will be, anybody can go find those data and rerun the analyses and see if their plots come out the same as ours. I see, movement in that direction over time, and I hope that will help with things like reproducibility. But I think the larger incentive structure, like how to move it, like the thing that I come back to and kind of puts my heart in my throat is that for prion disease, I feel that we're at this moment where the genetics are very clear, the tools are sort of, we have a lot of good tools. It's a really sort of ripe configuration for therapeutic development in some ways. And then on the technology side, we have not just ASOs, but other nucleic acid therapeutics, gene therapies coming along that are, sort of like modular, that you can sort of apply to different diseases with a certain amount of reconfiguration. And I don't know if these two things would have been brought together in our disease if we hadn't stepped up and said, this is exactly what we need to do, and we're just going to focus on that. There are a lot of smart people in our field, and I think they're doing all sorts of important work, but that particular thing of saying, are we ready for the first therapy? Do we have the tools? Do we have the information? Do we have the technologies? And if yes, let's go for it. Let's risk everything on it. I think a lot of smart scientists are drawn towards either, the basic science questions that are of enduring interest in prion disease, or technologies that are much more novel than ASOs, which have been, there's like 30 years worth of publications on ASOs. And, when we try to publish our manuscripts or apply for funding, we frequently get the feedback, there's nothing new about this. Like, we have known for a long time that if you lower PRP in a mouse brain, that's protective. And we've known for a long time that ASOs are theoretically usable technology, usable therapeutic modality in the brain. And so, you bring those two things together, and it doesn't exactly give you a lot of sort of like academic fireworks, but it's the thing that might make a difference for the patients. And I think I see other rare diseases where probably, the genetics are at a stage where they're ready, and there's a technology out there that might be at a stage where it's almost ready, but like, who is going to be incentivized to bring those things together if we don't think that that's a very impressive academic feat? I think some people think, well, that's a company's job. And for a common disease, certainly, I think a company would take a lot in-house, and maybe they would be scanning the world for different indications that match their technology, and they would do it all. But for a rare disease, what we've found is that you just have to assemble a whole platter of assets, and then try to interest the company in meeting you halfway. And so it ends up being this kind of hybrid, academic pharma undertaking. And I think we need to incentivize people to take on that academic half. and to make rare diseases developable, and to sort of see that as, a really amazing, a really amazing scientific contribution that is intellectually challenging as well as being, practically important. So I don't know how to change the sort of like relative status associated with these different kinds of things, but I guess I feel lucky that we were sufficiently motivated that none of these things have been able to deter us.
Science moves in mysterious ways, just like life in general. And basically, even though the arguments against working in rare diseases, as you lay them out and are known, seem quite clear. On the other hand, a breakthrough in this particular area of research and possibilities for translation could signal a chance for other similar diseases. We had the whole afternoon devoted to protein misfolding disorders. Would you perhaps give us a little bit of your perspective on how translatable the ideas might be to other diseases?
Sure. I think that's absolutely true. Despite the challenges of working in rare disease, I think, especially right now with genetically targeted therapies sort of coming online, I think this is exactly where we can look for sort of translation in our lifetimes. I think rare diseases often have the asset that they're genetically well-defined. And so instead of sort of trying different things and not knowing, was it the drug's failure or was it the failure of the therapeutic hypothesis? I think in rare disease, sometimes we have real clarity about the therapeutic hypothesis, and that's such an asset. And I think it actually increases your odds of success by so much. So I think that's a really amazing opportunity that rare disease offers. In terms of sort of the translatability of what we're doing, I think there are definitely other neurodegenerative diseases that are up against a really similar sort of problem, which is reduction of one disease-causing protein in the brain. And I think we've learned a lot from some of those diseases. We've followed progress in, for example, SOD1 ALS, which is ahead of us in terms of ASO development. And I think it's exciting to think that platform technologies like ASOs might be honed for a slightly more common disease, but then can be repurposed for rarer diseases that share, certain characteristics, the tissue of interest, the mechanism of the sort of direction of effect, the mechanism of action that you're going for. So I think there are definitely a lot of synergies right now. And we see it when we look across the landscape of different pharma companies. Like there's sort of a list of neurodegenerative diseases that pops up over and over. Huntington's, SOD1 ALS, tauopathies. And I think one thing that we can hope for is to get prion disease on that list, to have it be at a point where it is well enough established, all of the tools are there, the patients are there, the therapeutic hypothesis is solid. And now different people with different technologies look at it as sort of a place where they can establish a proof of concept.
From the perspective of a patient who, for a good portion of your life before, did not know you were a potential patient. You mentioned ALS before, and I was just reminded that today during the workshop, we actually asked the audience whether they participated in the ALS Ice Bucket Challenge. And quite a few did, apparently, from those who answered. The reason why I'm mentioning it now is because I was just thinking about the healthy population, the people who do not yet have any kind of a disease on their radar and are not really bothered by thinking about the prospects of that in the future. How much value do you ascribe to such actions that draw attention and increase awareness and can maybe mobilize people and possibly also increase funding, although I don't know. I know that there were some consequences of the ALS Ice Bucket Challenge in that sense as well, but probably not over a long term.
Sure. I mean, I think at a high level, raising awareness is great, and like whatever we can do in that direction is great. I agree that I think it was quite a successful fundraiser, but I also have the impression that it was like it was successful in that moment, and then the zeitgeist moves on to something else. So which is fine. I think if we could if we could harness sort of like public energy even for a short time, I feel like that would be powerful. It's we, you know, on a much smaller scale, we've run a nonprofit since 2012, since we got into this, and we raise funds to support our research through that vehicle. And I think what you said about it being sort of like a short-term burst has definitely mirrored what we've seen in terms of the different ways to raise money for research. We have been really fortunate to raise money through our nonprofit, but it's totally unpredictable. And I mean, I guess I could say that over time, as we've become more established, we've raised more, but you really can't count on it. You don't know what's going to come down the pike next year. You don't know if there will be, say, a global pandemic and how that will affect people's finances. Like it's all very, you take it as you get it. And that is another way in which we've come to, I think, in which we've come around to feeling like we need to compromise with the ways that things are done more conventionally. Because I think we've come to appreciate that the very traditional route of getting federal grants to support your lab does give you an amount of stability, like 5 years or whatever of stability, that it's really hard to get any other way. Even if you do have a viral ice bucket challenge, or like, you know, kind of your moment in the spotlight. So for right now, our lab sort of like straddles. We still have a lot of philanthropic support. We now have grant support as well. And I think they complement each other in an interesting way. So like, hopefully we'll have both going forward. But yeah, people say to us once in a while, why don't you just create an ice bucket challenge of your own? And I think there must be some interesting art or science to like what catches the public's imagination. I think everybody is always trying as hard as they can to go viral. And there's no saying when lightning is going to strike.
Yes. On the other end of the spectrum, when I started before speaking about the early life, everybody being young and mostly healthy, not thinking about diseases, and speaking now from a perspective of someone who is pushing 60, again, touching back on your topic before about engaging patients or people who are predisposed to become patients later, how important it is to mobilize them to participate in a number of different ways with a different motivation from the young crowd, obviously. I was quite amazed when I worked in prion research back in the day to see how motivated people with another genetic prion disorder, Gerstmann-Streusler-Scheinker's disease, were to actually participate in research that did not promise anything other than maybe a small step forward in advancement of early diagnostic procedures. And I think it's valuable pointing out that moving away from rare genetic disorders, all of us are on trajectories that probably lead to some sort of disorder of the brain if we just live long enough. Would you help us formulate some suggestion as to how to motivate the wider public to maybe take a more active role in participating in research or aiding the work of scientists in some additional way?
Oh, gosh, no, that's a great point. And I wonder if, I've wondered if part of it is that here in Boston, I look around and we live a very age stratified life. Like we tend to interact with people who are our age or people who are between the ages of 1 and 5. And I think you lose something in that sort of social configuration. You lose the sort of like generational perspective. And I think we definitely know a lot of people who are like young and healthy and seem like they feel bulletproof. But absolutely what you say is true, that I think neurodegeneration is something that we should all be thinking about. And it would certainly help our, I think it would help our cause to the extent that people were thinking about it before it happens. It's not a pleasant thing to think about. It's not an easy thing to think about, but I think, prevention hinges on an awareness of risk. And that's true in the very acute sense in our genetic prion disease community. But I think there's sort of a broader awareness of risk, that applies to everybody. As you say, should we be lucky enough to live long enough? And that's where I think there's like a larger conversation to be had about where neurodegenerative drug development should be moving, because I think if people stopped and thought about it, I think anybody who really thought about it would say, of course, I want to prevent neurodegeneration in myself or my spouse or my parent, and not just be trying to treat it and stabilize them once they're already so sick that we're taking them to a neurologist. So I think our message actually has like, it does have this like broader implication. And I think maybe now people are becoming like a little bit more aware of brain health and a little bit more aware of steps you can take to stay healthy. But there's this other step of, beyond prion disease for other dementias that, pose risk to a lot of people. How can we, and maybe it's a longer road for some of them because the pathology is not so clear and the genetics are not so clear and we're still piecing it all together. But how can we eventually move towards sort of a preventive paradigm in those diseases too? And I hope that society will sort of grapple with these questions, because it's going to be important not just for, designing clinical trials and sort of calculating risk benefit as we enroll people in trials, but also for things like reimbursement, where, eventually what this all means is we're going to be asking drug companies to pay for drugs to keep people healthy who look healthy today. And maybe they're going to say, no, you're healthy and we pay for drugs for people who are sick. But I think we as a society need to do the calculus. Is that what we want? It's probably not what we want for ourselves. So it's probably not what we want more broadly as policy.
Yeah, many challenges that we face as a society, I guess, like you pointed out. We are going to talk about the concept of brain health and how to possibly use it in promoting healthy lifestyle choices tomorrow during the remaining day of the conference. And it is one of the directions in which Synapsa, the society that organized this conference, has been active in the past couple of years, trying to really promote the idea that there's just this one brain that we all have, and it's uniquely and in more direct ways connected to just exactly who and how we are, to all our potential in terms of learning and ability to be the people we are. And we need to try to individually take better care of it throughout the lifespan. And also we need to probably, as you pointed out, think about some policies that might perhaps make it easier for people to try to do the bit themselves to take care of their own brains better. We did have some specific questions related to research. So if it's okay, maybe I go and ask those. Have you looked at any neuroimaging biomarkers in your research?
So we don't do a lot of neuroimaging. There's a literature on sort of diagnostic imaging in prion disease. And we know that MRI can detect particularly sporadic CJD. It's how a lot of people are diagnosed. There's also a little bit of indication that PET imaging can detect hypometabolism in the thalamus, particularly for the FFI subtype. So there is clinical imaging in prion disease. What we don't have is an indication that imaging could give us a signal before symptoms. And where people have looked at this, it seems like the imaging changes tend to basically coincide with symptoms, whether it is by MRI or by PET. At most, maybe a couple of months before symptoms, you might see something, a few months. So we haven't put a lot of energy so far into doing imaging on our carrier cohort, just based on what's out there. The other thing we get asked often is whether we have a PET ligand that we could use to, say, measure PRP abundance in the brain. Unfortunately, we don't. We don't have a small molecule that specifically binds PRP. So that is a tool that is yet to be developed.
Another specific question deals with what would the design be for the actual study? I guess this is relating to the ASO with Ionis.
Sure. So I can't speak to their exact clinical plans, but I can say what we've advocated for, which is that in parallel, or in addition to a symptomatic trial where the design is probably less difficult to imagine, that we would have a pre-symptomatic trial where we enroll people with high penetrance mutations and we treat with ASO or placebo in probably in various doses of ASO, and measure prion protein in CSF and see if there is a dose-dependent decrease in prion protein in CSF. And also measure, of course, like safety markers across the board. And the vision would be that from a data package like that, we would get, under the accelerated approval pathway at FDA, we would try to get provisional approval for carriers to go on drug. So that's step number one, is if we can show that prion protein is going down, is that convincing based on everything we know that this is having a protective effect? If yes, can we get people on drug sooner based on what should be really a pretty swift study? From there, accelerated approval comes with the condition that you follow people in the long term and confirm clinical benefit. And I think doing a sort of pre-approval study where you follow people to endpoint, I earlier made the case that it just simply would be impossible for a company to invest in that. But I think if we're talking about a post-approval scenario, then people are on drug, the company is being reimbursed for the drug, and they just have a responsibility to monitor people's eventual outcomes. I think that is a more realistic ask. And the hope is that the impact on age of onset would be significant enough that we would see it when we compare it to a database of natural history controls.
Well, I will be a bit provocative here, because I thought that I remember Ionis was ready to go at sometime during this year, and now has the goalpost been moved a bit?
It has, and
It has.
I think I always have to remind myself that these things are just not unusual at all in drug development.
Okay.
But basically, so what they shared publicly is that they pivoted from the first human clinical candidate that they had imagined moving forward into the clinic to a different human clinical candidate that they think they like better. Hopefully this improves the program in the long run, but it does mean that we lost about a year off the projected timeline, which is really disappointing. There's no doubt about it, but I hope it helps us do better trials in the end.
When you mentioned it before during your talk, there wasn't even a slight indication of disappointment, and I paused and thought, wasn't it supposed to be this year? So I decided to ask you. Yeah, no, it's a difficult process, but I guess the optimistic slide that you shared with us with your two beautiful children essentially speaks for itself. I mean, even though we criticize science and many issues related to how it's done these days, it is a triumph of science that we've come this far, and that you also have those two beautiful gems.
Yes,
around
absolutely.
you. Right. So even if it takes a bit longer to get it right, we're all for it, I guess.
Yeah, I think that's right. It is fairly amazing that we've been able to benefit from sort of genetics, clinical genetics in practice. And I think the IVF PGD experience itself was never a straight line. It was like lots of ups and downs, just like drug development. But we were patient, and we got a really miraculous outcome in the end. So I have to hope that if we bring that patience to bear on our work in the lab as well, good things will come of it.
Let's end on this note, because I think we have drained you of some energy today, because this was a long session. I speak on behalf of my team here immediately next to me, and also I think in the name of the audience that stayed with us, when I say that we are really grateful and rooting for science and your work to advance with great strides. Thank you very much, Dr. Vallabh. Sonia, if I may, and take great care. Hope to see you in a few years presenting on the progress of the work that you've described as future.
Oh, I hope so too. I hope the story just keeps getting better. Thank you so much for having me. It was really a pleasure.
Most welcome.